हॅलो एव्हरीवन, नमस्कार बाळांनो. आता पुढचा पार्ट आपण बघूया की ज्याच्यामध्ये आपल्याला एम्ब्रियो डेव्हलपमेंट बघायचं आहे. सुरुवातीला आपण एका व्हिडिओमध्ये पाहिलेलं आहे आणि लेक्चरमध्ये की एम्ब्रियो डेव्हलपमेंट डायकॉटमध्ये कसा होतो, तसंच आता मोनोकॉटमध्ये कसा होतो. दोन ग्रुप आहेत, नो डाऊट. अँजिओस्पर्ममध्ये फ्लॉवरिंग प्लांटच्या एक डायकॉटिलिडॉन आणि दुसरा मोनोकॉटिलिडॉन. डायकॉटचा एम्ब्रियो डेव्हलपमेंट आपण पाहिलेला आहे. आता मोनोकॉटचा एम्ब्रियो डेव्हलपमेंट कसा आहे तो आपल्याला आज इथं पाहायचा आहे. हे ज्या वेळेला आपण पाहतो त्यावेळेला आपल्या असं लक्षात आलं पाहिजे की डायकॉट आणि मोनोकॉटमध्ये फारसा फरक नाही. अप टू द क्वाड्रंट अँड ऑक्टंट स्टेजपर्यंत दोन्हीही एकसारखे आहे. तिथून बाकी काय आहे की प्रत्येकाचं डेव्हलपमेंट ही सेपरेट होते. डायकॉटमध्ये 2 कॉटिलिडॉन्स फॉर्म होतात आणि मोनोकॉटमध्ये सिंगल कॉटिलिडॉन फॉर्म होत असतं. तसं आज आपण इथं पाहूया एम्ब्रियो डेव्हलपमेंट or female gametophyte development in the monocot plant angiosperms. We look at here, the zygote is the primer cells, first cells by which division the cotyledons are formed. These cotyledons are called as monocotyledons, and monocotyledon structure is very different as compared to the dicot. After the zygote formation and successive fertilization, zygote undergoes division, transverse division first, and gives two-celled structure in the B side. We look at here this B diagram. One is called as CA and another is called as CB. CA means apical cell and B means basal cells. Apical cells different and basal cells different. Functions are also different, but they are just like the dicot. Then again, these two cells are undergoes first transfer division by CB and forming CI and M, while CA is again longitudinally divides to form CA and CB. So it looks like two cells, elongated one, two cells perpendicular. So each of them shows inverted T-shaped. So this inverted T-shaped pro-embryo is developed. This is most important in the case of monocot development or female gametophyte in the monocot. Then afterwards what happened? The upper cells are again divided N to N dash. Lower cells form two tiers. One is M and another is Q. Then this division is again visualized more in the next stage to forming a quadrant stage, and quadrant stage into the octant stage. This quadrant stage and octant stage as same that of the dicot plant, and we look at here in E diagram how it forms. The basal part of the embryonic cells divides two tiered. One is L and another is L dash. Easy to identify. L is light in stain and L dash is dark in stain, and remaining suspensor cells just like in the dicot, they are M, N, O, and terminal is a P. Then again the division has been takes place in the embryonic cells and forming the different types of the cells, dermatogen, then epidermal initial cells, then another one use peribleme, then pleurum of the root, and root cap where hypocotyle, and this hypocotyle responsible for formation of the plumule. Then the division is continuous embryonic cells and forming a single cotyledonous structures, a distinctly showing L dash, L, M, N and N dash. N dash is called as suspension cell, which is very small, and this suspension cell deep into the nuclear cells of the nucleus, and then absorbs the nourishment and supply to developing a single cotyledonous embryo. So this is about the diagrammatic representation. As we're looking about, there is no much difference between the dicot and monocot embryo development regarding the early cell division of the proembryo, which I already discussed about proembryo, that is the inverted T-shaped proembryo. However, the mature embryo is so different in that case, and this is a typical example from Luzula forsley under the family Mucaceae. This is from the monocot. It is a simple type of embryogeny and described by Sages in 1923 C. Zygote is divided into two cell proembryo and forms C and CB. This proembryo divides by longitudinally wall to produce two juxtaposed cells, and little later the basal cell CB divides by transverse wall to produce the two cells CI and CF at the upper side. In the next stage, what happened? The two cells of the tires C undergoes another vertical division at right angle to the first and gives rise to quadrant stage Q, and the M also divides by longitudinally and longitudinally with wall formation to give juxtaposed cells. CI divides transversely to form two cells N and N dash. By further divisions, the quadrant partition into two portions L and L dash, of which L gives to the lower part of the single cotyledon and L dash gives the upper part of hypocotyle and plumule of the embryo. The remaining tires M gives rise to the periblem and part of the root cap, while N gives a remaining part of the root cap, while N dash gives to small suspensor cell, that is the O and P. So there is immediate differentiated of epidermal initials after the quadrant stage, but in another angiosperms it shows after the octant stage. Only in this case, quadrant after the quadrant stage differentiation takes place, but majority of the cases in the another groups and another plants of the monocot angiospermic plants, it shows a very different one. Thank you.